The overall goal of this methodology is to create immobilized bioactive spatial protein patterns which can be used to study cellular responses.
This method allows for recapitulating in vivo signals using biomaterial strategies. This technique allows for accurate mimicry of certain signaling motifs that cannot be achieved using standard culturing methods, such as sequestered growth factors, cell-cell signaling, and spatial-specific biochemical cues. This protocol is significant to existing methods as it provides a simplistic method for adjusting substrate stiffness and protein patterning. While this method can further tissue engineering and regenerative medicine technologies, it can also be applied to study other systems such as tissue development and stem cell fate.
To begin, prepare stock solutions for the major hydrogel component, polyethylene glycol diacrylate, the photoinitiator, lithium phenyl 2,4,6-imitrimethyl benzoyl phosphonate, and the ECM protein fibronectin under sterile conditions as described in the accompanying text protocol. Filter sterilize each solution using a 0.22 micron syringe filter prior to using or storing the individual aliquots. Then, wrap the PEG-DA solution with foil to protect it from light. Wrap the photoinitiator stock solution in foil to protect it from light. And store the prepared solution at 4 degrees Celsius for up to several months. If fibronectin stock solution is frozen, allow sterile aliquot to thaw on ice for several hours or in 4 degrees Celsius overnight.